Hello, Claire. Hello, ladies and gentlemen. Thank you for finding the time to attend our webinar. This is the first time we are doing this, and we have planned this since last year. We were hoping to do this December, but that was moved to January, February, but finally we're all here. So maraming salamat again for coming and attending our meeting. First of all, just a second, let me fix my... Can you see already my presentation now?
Yes, sir. Though hindi po siya naka-full screen.
Hindi naka-full screen. Just a second. Okay? Is it okay?
Yes po.
Okay. So, yeah, let us start. Just a brief backgrounder, how we got into this business anyhow. Because I come from a totally different field. I'm an aquaculturist by profession, but I decided to move to a different field. It's a very long story, but in any case, this field that I wanted to go in was natural products. And I had very little knowledge of this field, so the first thing I did was to look at the value chain and see what are the opportunities I can go into. And I quickly realized that ingredient manufacturing is pretty much a non-existent industry in natural products in the Philippines. And when I say ingredient, I mean those that are processed ingredients. So the very first product I went into was medicinal mushrooms. DOST6 was very much helpful in the growth of our company, and there was a DOST setup program in 2006 where we were able to borrow funds based on a 3-year interest-free loan, and we decided to purchase our first R&D equipment, which you can see here. It's a spray drying granulator, and this pretty much became our platform for making ingredients during the first 5 years of our operation. Later on, we realized that using a spray drying granulator has its limitations, so we decided to invest on the real stuff, which is a large extractor. You can see on the left side, that's a 500-liter extractor, then a concentrator, 500 liter, and on the right side is the spray dryer, which is rated at 25 liters per hour. So pretty much we pioneered the production of spray-dried botanical extracts during that time. And little did I know that DOST PCHRD was planning to launch the Tuklas Lunas program, which is one of the flagship projects of DOST. So it was started in 2012 with a vision to produce world-class medicines derived from Philippine biodiversity, leveraging on local expertise. And these are the partner institutions, and I think pretty much everybody is here. And there are now 4 from industry, and Urbanex was fortunate enough to be the first private sector to join the program in 2013. And these are the products that PCHRD is helping us right now develop: herbal drugs, herbal supplements, and there's actually a relatively new program, which is functional foods. There are still other markets like cosmeceuticals, animal care, plant care, but these are not in the area of PCHRD. Just to show the relationship, herbal supplements and functional foods are grouped together actually, and they serve the preventive medical approach. Whereas herbal drugs are grouped with OTC and prescription drugs, and they are treatment for sickness or injury. So as you can see, herbal supplements is actually a combination of food and drug. These are the products that we have produced so far. Tawa-Tawa was the first product that we launched in 2019 during the dengue epidemic. And this is the first product that bears the Tuklas Luna seal. And these are 5 other products you can see on the lower left. And we are talking right now with a couple of pharmaceutical companies. Hopefully they would like to commercialize the output of our research. One thing I realized as we went in the we worked on the Tuklas Lunas projects is that the ingredient development and scale up was not really in the program because from my understanding, it is really the industry that should do this. But this is easy to do for powdered herbs because you really don't need any specialized equipment for this. But for spray-dried extracts, very few companies have the proper facility, and Pascual is one, of course. So we have assisted some of our colleagues by producing for them the extracts for the last couple of years. Just to share with you some of the common problems we encounter with working on crude extracts, which is basically the platform of our supplements and herbal drugs. There is really a problem of high hygroscopicity for most extracts, poor flowability, caking, poor shelf life, and almost always you have to use high level of excipients which dilute the active ingredients. And for functional food applications, there are limitations like caseiness, astringent taste. And another problem encountered in scaling up is that change of scale is not symmetrical as rightfully written here by a Nobel laureate in physics, Richard Feynman. It is not true that if you build an apparatus and then build another one with every part made exactly the same, of the same kind of stuff, but twice as big, that it will work in exactly the same way. So it does not come as a surprise that as we try to scale up our 1 liter extracts, we noted differences in quality. We were fortunate that in 2020 we were awarded the first BIST loan, which is it's an interest-free loan for 3 years, and the money has to be used for R&D. And so we decided to acquire several equipment for research on ingredient production, and we decided that we realized that we were in a unique position to help accelerate the growth of the herbal research, particularly in the Tuklas Lunas program. So we decided to open up our facility to our colleagues in the program. So in December 1, 2020, we launched ARIL. This is one of the primary work we're doing right now, which is using the equipment we purchased from the Business Innovation through Science and Technology loan. And this is the removal of nuisance compounds, which I mentioned earlier, is a problem of crude extracts. These are oils and waxes, particulate solids, starch, pectins, organic acids, sugars, and tannins. And they need to be removed or reduced, otherwise they're really going to affect the stability of the ingredient and eventually of the product. So we set up different equipment in our pilot scale facility. Here's a list of some of those equipment, and as you can see, we have a gradual scale up from 0.5 to 5 liters for extraction in a rotary evaporator to 10 to 20 liters, and finally to 100 to 300 liters. So the ARIL is purpose-built for the development and scale-up production of standardized ingredients and herbal dosage forms. It's equipped with advanced pilot-scale manufacturing equipment for validation and enhancement of laboratory-scale technologies. As I mentioned, it's funded by BIS, and it's supported by a multidisciplinary staff consisting of chemical engineers, chemists, pharmacists, food technologists, and even mechanical engineers. So we're open to research collaboration and contract manufacturing services to all natural product researchers from the academe and government. The key objectives of ARIL are optimized manufacturing process for highest AI or active ingredient yield and lowest production cost, to reduce nuisance compounds in food extracts, develop scale-up production technology, um, standardize quality and meet GMP requirements, customize ingredients and customize ingredients according to client requirements. So basically, this is the flow of what we do in scaling up and product and process development. First, we start with a pre-feasibility study to determine if the product will produce a competitively priced product in the market. We have plenty of data as we are a commercial manufacturer, so we can have a good estimate of how much the product will cost if it will be scaled up. If the cost is uncompetitive, then we have to review the production strategy and market positioning. If it's competitive, we go to the next step. We check the abundance of raw materials. If they're abundant, then we proceed to the next step. If not, then we have to address the supply issue first before proceeding any further. So we go to standardization, raw mat processing, number 3, and then we go to API manufacturing or active pharmaceutical ingredients. So we check here if the bioactivity is retained and the API is stable. If not, then we have to address that problem, improve the manufacturing process, and excipient formulation. If it's once the stability is addressed and bioactivity is there, then we go to the dosage for manufacturing. If it's stable, then we go to IP application and finally FDA registration. So this is pretty much really the process that we go through. More or less, this is going to take 15 months. We try to squeeze in as much as we can, not possible. Maybe 13 months, but realistically 15 months to do a real honest to goodness scale up process. Just to give you some idea, the typical MOQ for toll manufacturers is around 250,000 capsules or tablets, usually capsules. So this will require approximately 50 to 75 kilograms of extract, and 50 to 75 kilograms of extract will require 200 to 400 kilograms of powdered herb, which will require another 1 to 5 tons of fresh herb depending really on the nature of the herb. So as a rule of the thumb, 10 kilos of extract will require 100 kilos of powdered herb and 1,000 kilos of fresh herb. So here you can clearly see it's no joke to commercialize. If you can't even produce 1 ton, then there's really no point in thinking of commercializing your product. So the estimated cost for scaling up, I mentioned this on the 2019 NTSP Congress. I mentioned 0.5 million to 1.5. Now we're pretty much computed properly. It's around 730,000 to 1.43 million, depending on the state of the research done and the difficulty of working with the herb. I would like to share to you some figures just to give you an overview of the type of work that has to be done when you do ingredient development and when you assess its feasibility. So let us look at, so these are the cost of AI, active ingredient dosing per day, of 5 different herbs. This is just active ingredient per day. This is not the finished dosage form. And this is usually about 3 servings. Divide this by 3 servings. No? So again, this is not the finished dosage form, but this is a good indication already of how the economics of the product will turn out. So if you can see on the leftmost side, on the rightmost side, sorry, the cheapest to produce is herb E. And if you look at it, why is it so cheap to produce? First of all, because it's just water extract. Your solvent is just water. And secondly, your dosing. Okay, the dosing, by the way, here are based on published literature. No? So we did not establish this dose. It just so happened that the herbs we're working with are kind of known, so there are recommended dosings. So the dosing is relatively low. So that is what produces a low cost of 1.54 pesos. If we go to herb B, you can see that what brought it to a relatively low cost, considering that it's ethanol, is that the raw material cost is low, only 450 pesos per kilo. And the amount of raw mat needed to produce 1 kilogram of spray dried extract is relatively low also, 7.39 kilos, while some herbs require 11 plus kilos. Don't bother on the rightmost, which is the fresh weight. That's why it's very high, 29.41 kilos. But the price also is very low because it's fresh herb. So you can see here that an interplay of factors have a direct effect on the production cost of your AI or the requirement of your AI. On the other hand, if we look at the most expensive product that turned out, well, because the raw material is expensive, and although the marker compound in the extract is high, 17.25, it's the highest of all the herbs being compared here, the dosing required is also very high, 150 milligrams per day. So you can see that you have to first be sure that the raw mat cost is reasonable, the lower the better. If you can use less ethanol, more water, maybe still ethanolic, but reduce it from 80% to maybe down to 50%. If you can establish that you can produce the same, you can extract the same bioactives, then do it. And so when you're selecting palang, if you have several plants to work that you can select, these are factors that I would look into and I would prioritize. Because even though how nice your bioactivity is, if your product becomes very expensive, in the end, the industry cannot take it because it's just not feasible for them to market it. I mean, there are so much options for them of active ingredients they can use. Why are they gonna bother themselves with an expensive ingredient? So my rough estimate, if these products are manufactured commercially from a toll manufacturer, this will cost for an MOQ of 1 million, minimum order quantity, this will cost around 350 to 550 per unit, depending on the herb. So if you choose your herbs properly during the selection process pa lang, as I showed you the process flow that we do, you can be more towards the 350. And if not, you will be towards 550 or probably even more. No, there are some that I've computed at 10 pesos. So before you invest 2-3 years of your career on a particular herb, it's important to know early on if it really has a chance to be commercialized. So I think because I don't have really much time, we have a reasonable, fairly long open forum later on. I just like to show to you my team. They're, as I mentioned, chemists, pharmacists, ChemEng, so on and so forth. So this is my team, and these are my kids actually who have been helping me since my mushroom research days. Aidan, who will be talking after me, is a molecular biologist, and on the right, Tion is a chemist. So thank you for your time. Later on, we will have an open forum, and I'd be happy to share to you all our experiences in Ariel. Thank you again.
Thank you for the very informative and interesting presentation, Sir Philip. I am so happy to see this initiative coming from the herbal industry in the country. As we all know, Philippines is very rich in natural resources, and having this technology will help us scale up many of the significant findings of our researchers from the academe and other institutions, especially those that are considered to be low-hanging fruits. The product of your years of readings, experience, and all the trial and errors are now coming into shape. Identifying the common problems encountered when working with crude extracts and finding the solution to overcome those problems can be of great help to our researchers. It is always good to hear from the industry, especially on the aspects of marketability. Thank you for sharing to us or for giving us a deeper understanding of the production cost. That was very helpful, especially for those who wish to market their finished product. So again, thank you for starting all of this improvements, Sir Philip, and I'm sure more to come from Tion and Aidan. Thank you, sir. Now, for sure, many from our participants right now are looking at the possibility of collaborating with Herbanex. Perhaps our participants are now having some questions in their minds. Please be reminded that all your questions will be entertained during the open forum session after the last presenter. But you can already type your questions in the Q&A panel, as there is a representative that will collect your questions and it will be read during the open forum session. Thank you. Now, let's move forward to our next presenter. He is a molecular biologist by training and a natural products researcher in practice, who graduated from UP Diliman in 2018. He works as the research and extension head of Herbanex Incorporated, managing existing partnerships between the company and its research project stakeholders. He is also currently taking his MS in biotechnology at the University of Melbourne, and in the near future, hopes to be able to incorporate biotechnological approaches to product and process development in the herbal industry, allowing for the building of key capacities in industrial biotechnology in the region. Let us welcome Mr. Phil Aidan Cruz to deliver his presentation on Understanding the Bioactivity of Herbal Extracts. Mr. Phil Aidan, the floor is yours.
Thank you very much for that introduction, Ms. Claire. So I'll be sharing my screen now. Yep. All right. So all this talk about the herbal industry, I think it really boils down that first we need to have a common understanding of where bioactivity in herbal extracts come from and why we're going to be using herbal extracts in the first place. So this is going to be the purpose of my talk. I know many of you, or almost all of you, are actually collaborators in the Tuctas Lunas program, so you pretty much have a good understanding of this. So apart from just reiterating this, I guess it's also good to have a common understanding for all of us. So let me begin first with an introduction about where bioactivity in plants arises from. So throughout most of their evolutionary history on Earth, plants have developed numerous means to cope in a hostile environment that's filled with herbivores and different pathogens. You know, plants, they're sessile creatures, they don't move. So instead, they have become very creative in creating a chemical arsenal of compounds to do their bidding. So secondary metabolites are molecules synthesized by plants that aid them in their survival. And they play numerous roles for plants, like UV protection, as an antimicrobial agent to communicate with other plants, to prevent consumption by herbivores, and to form symbiotic relationships with microorganisms in their environment. So we might think as humans that we're so special that plants have created these compounds for us, but in fact, we only share a very small blip of our evolutionary history with plants, which have existed on Earth long, long before, together with insects, for which many of them have specialized their chemical biology to interact with. So many of these compounds produced by plants are in fact for interaction with insects and other animals. So an example here is anthocyanins in fruits and in leaves. So we can see here the compounds that make strawberries red are actually present because it's intended to attract animals that can consume the fruit and disperse its seeds. And the red color here you can see in the leaves of this plant are intended to absorb UV light from the sun to prevent mutagenesis in the cells. So when humans arrived as early as 1500 BC, in one Egyptian document called the Ebers Papyrus, has described that we have already been using whole herbs and herbal preparations to treat various ailments and diseases. But it was not until the year 1805 when this German apothecary named Friedrich Serturner isolated the narcotic principle of opium, which is called morphine. And this paved the way for the advent of the field of pharmacology in the 1800s. So, with the advent of this chemically defined small molecule drugs, it would lead one to assume that herbal medicine would become obsolete because you can just isolate the active compounds. But the thing is, this complexity in herbs is what makes them just as effective, and that is the reason why, as a field, herbal medicine is here to stay. So to gain a better appreciation of how this efficacy in herbal extracts happens, we have to take a look at them from a pharmacological perspective. So in herbal extracts, more is more. And despite our long history with herbs, we have a lot to know about the mechanisms of their action. And the complexity of and variability of herbal medicine is actually what makes it difficult to associate the activity of these extracts to a single chemical entity. And this is counterintuitive to the goal of drug discovery, which is to discover a single active compound. However, herbal medicine is popular because of its efficacy. Some herbal medicines can even reach up to the same level of efficacy as single molecule drugs. And this is, in fact, a direct consequence of their complexity, wherein interactions between the different constituents in the plant also interact with the complex nature of different diseases. And this is what makes them effective. So when I say complex nature, I mean, take a look at this diagram. This is the pathway for asthma. And I got this from the database, the Kyoto Encyclopedia for Genes and Genomes, or KEG. And you can see that asthma involves a whole cascade of different inflammatory mediators, such as interleukins here. And, a lot of products on the market, like biologics, they're aimed to target single receptors here, like interleukin 4, interleukin 6. Whereas when you have a herbal extract which contains numerous components, it can address different interleukins because they have different binding affinities for each. And compared to just addressing one in the pathway, which will just like block one part of the cascade, you can address the majority of the proteins involved in the cascade. The same goes for this pathway for rheumatoid arthritis. You can see that this is even more complex than asthma. It involves factors like angiogenesis, bone resorption, joint destruction, and inflammation. And as a disease, rheumatoid arthritis is still not as well characterized, and there's more to understand about it. There's also the factor of drug resistance in bacteria. So bacteria have evolved numerous ways to evade single molecule drugs, and herbal medicine has proved to be one of the effective ways to actually treat it. So an example is in this bacterium, which has evolved the NOR-A efflux pump, which can remove quinolone antibiotics from its system. So this is a single molecule drug. And by treating bacteria with quinolone antibiotics together with Scutellaria bicalensis extract, you can have compounds that inhibit the NOR-A efflux pump, restoring the antibiotic activity of quinolone antibiotics. So you can think of it as a spear versus a net analogy. So a single molecule drug has a single target or just a few targets, and you can effectively kill the target or capture the target using that single drug. Whereas herbal medicine can act more like a net, and in turn you can capture more fish or more targets rather than using a single molecule compound, because it contains a numerous array of compounds. So compounds in herbs interact in a multitude of ways. They can be either additive, synergistic, or antagonistic. And there have been different models developed by scientists to explain these interactions. There's the Bliss independence model and the Lowy additivity model. We don't really have to go through that very much in this discussion, but just so you know that interactions in herbs are not always 1+1. And this is exactly the reason why efficacy in herbal extracts can actually reach drug-like levels. So this is one example of a possible synergistic interaction in this plant called Astragalus membranaceus in its root extract. So a systems biology approach to understanding its chemistry, they were able to find 44 compounds, which is about almost 50% of all its compounds which are known, that show potential activity against cardiovascular disease. So you can imagine that there are compounds here that act as antioxidants. There's also ones that reduce the production of VCAM1, which is a cell adhesion protein important for inflammatory cells to travel through blood vessels. It can also increase the N-guanylyl cyclase pathway or improve blood flow, can improve calcium transport in the sarcoplasmic reticulum, and can also reduce blood pressure. So this is gamma aminobutyric acid. And you can imagine that all of these compounds acting on a single pathway can in fact produce a much more profound effect as compared to all of them just acting as like reducers of VCAM1 or antioxidants. Another example of this one is antagonism. So we have echinacea endophytes, and they were wondering why echinacea extracts, which are supposedly anti-inflammatory, exhibited pro-inflammatory properties. And what they found out was that in the polysaccharide extract containing extract of echinacea, they found an abundance of endophytes which produce lipopolysaccharides, which are pro-inflammatory polysaccharides. So since they're similar chemically in nature to echinacea polysaccharides, once the researchers removed the polysaccharide containing fraction, they were able to restore anti-inflammatory activity in echinacea endophytes. So there's in echinacea extract. So there's a lot of consideration to take, especially in terms of compound interactions. So we just talked about compound interactions, so we need to understand just how much chemical diversity there is in herbal extracts and how these compounds give rise to their bioactivity. So herbal extraction involves separating the medicinally active portions from the inert portions of herbal raw materials. And each extract you take of a plant is essentially a snapshot of the whole plant metabolome obtained using chemical solvents and visualized using chemical analysis. So depending on the solvent, the properties of your extract will definitely vary. And many factors should be considered in extraction, including the process itself, the solvents you use, and the compounds of interest that you're planning to get. So this one book by Pulok Mukherjee in 2019 is a good reference. It gives you a good overview of the different compounds you can find in herbal extracts and their quality control and evaluation. So just going through some of the most important bioactive compounds you have in plants. So these are secondary metabolites. We have like alkaloids. So these are classified as nitrogen containing. And in plants, they function as part of the plant's defense system. So alkaloids consist of some of the most important compounds we have discovered so far, like caffeine, cocaine, vinblastine, and quinine. And in humans, they serve a different effect. They can be hallucinogenic, stimulant, or cytotoxic. And the main reason for this is just because plants and humans are very different biologically, we all do share a similar biochemistry. All of life shares a similar biochemistry. So for compounds that would act strongly on a certain protein in a different organism, we might expect interactions to also occur in humans as well, despite the differences in biology. So alkaloids are soluble in acidic water or polar organic solvents. And yeah, those are the examples I mentioned earlier. We also have volatile oils. So these are what give plants their characteristic odor. And they are small compounds used by plants to deter herbivores, attract pollinators, act as antimicrobials, and communicate with other plants. So scent is a very powerful tool in the plant kingdom. And volatile oils can be classified based on the compounds they contain and extracted using solvents like dichloromethane or chloroform, or using water by steam distillation. Just something important and some noteworthy things to show. Important compounds isolated from volatile oils include artemisinin and Taxol, which are very important drugs, artemisinin for malaria and Taxol for cancer, and other compounds like forskolin and asdirectin. We also have fixed oils and fats. So these are compounds that are consisting of triglycerides, of fatty acids, and some sterols. In plants, they can be an energy source and regulate water loss. But in people, these have been found to have a lot of nutritional and cosmetic applications, especially in the pharmaceutical industry. So we all know about the recent studies on coconut oil as a treatment for COVID-19. Flavonoids are some of the more popular compounds we can find in plants. They are water or alcohol soluble. And in plants, as I mentioned earlier, they're used to protect from UV-induced damage because they are free radical scavengers. So in plants, they can attract pollinators or attract seed dispersers by making fruits red and making flowers colored. But in humans, they also serve the same purpose as antioxidants, which is why it has become very popular nowadays to sell products containing high antioxidant levels. So yeah, they are classified by having a flavan ring system, which you can see here. It's a very common pattern in the different compounds shown. And popular flavonoids include silymarin and rutin. Tannins, these are a popular term when you talk about wine, and they are a class of alcohol and water-soluble compounds. So tannins can be classified depending on their size and structure. And in plants, they function to protect from bacterial and fungal infections. However, in people, they would serve similar purposes as antimicrobials, antioxidant compounds, anti-inflammatory, and immunomodulatory compounds as well. So gallic acid is a popular example of a tannin. We also have peptides and amino acids. So in plants, we know that we all know the definition of a peptide and a protein, but in plants, these small peptides, they function mainly in plant signaling and in plant defense, as plants don't have as complex an immune system as us. So we have a very popular peptide now here is called Cyclotide, this one. And this has been isolated from Clitoria ternatea or the butterfly pea, and it has shown to have good antimicrobial properties. So this has actually been commercialized into an agricultural product. Other modified amino acids like 5-hydroxytryptophan in plants are used to scavenge free radicals, but in humans, they actually serve as biosynthetic precursors for serotonin. So eating plants containing 5-hydroxytryptophan might make you happy. And one of the last major examples we have are steroids. So plant sterols, they can be either free or glycosidic in form, and they are interesting in that they mimic humans' cholesterol and other human hormones in structure. So steroidal saponins like this one, diosgenin from yams, are analogous to sex hormones like testosterone, and can also have similar and are being studied nowadays if they exhibit a similar effect. Cardiac glycosides, on the other hand, like digitoxin and cardenolide, they're therapeutically important because they are able to increase systolic contraction and contractility in the heart. So they might be able to restore heart function or in fact arrest heart function and cause heart attack. So we just discussed so many different compounds. There are actually a few more classes mentioned in Mukherjee's book, but those ones you mentioned earlier are some of the most important. And a lot of consideration needs to be taken when you develop a process for extracting these bioactive compounds from plants. Firstly, you need to know what bioactive compound you're looking for, what family of compounds does it belong to, because that would dictate what solvent you're going to use for extraction. So this figure, a very handy figure here, you can see that these are the different solvents classified based on their polarity. And these are the general solubilities of different families or classes of compounds in those solvents. So if you take a water extract, you wouldn't expect to find much oil or much essential oils in your extract because they are not very soluble in water. And we're just talking about extraction here. Extraction is one of the most important phases of your process, wherein you retrieve the bioactive compounds from the plant material, but it's only one portion of the whole process and one of the considerations you need to take in process development and scale up, because there's also factors like the nature of the raw material, the pre-treatment of the raw material, like purification, standardization, formulation, etc., and has to deal with a lot of experimental design, statistical modeling. It's not as simple as developing a lab scale process and then automatically that's the whole that process is going to be the one that smoothly transitions to large scale. So Andrea and Herian will be discussing these factors also later in their presentations. But I hope this figure gives you a good overview of that. And So as facilities like Ariel, we hope to be able to assist you in the whole process development, in the whole challenge of process development from lab scale to scale up. And and from this presentation, I hope you were able to consider that there are a lot of things you need to take into account. It's not as simple as extracting compounds using water or just ethanol, because you have a whole range of other solvents as well, depending on your target compound. Yeah, so that's just the whole extraction part in this diagram. So I hope you'll be able to scale up with us in Ariel, and we look forward to collaborating with you. Thank you very much. These are my references. Thank you.
Thank you for that very informative and comprehensive presentation, Sir Aidan. Our minds right now are refreshed about the bioactivity of different metabolites found in herbal extracts. Aside from the therapeutic effect of those metabolites, it is also worthy to note the interactions of constituents present in the extract, which may produce additive, synergistic, or antagonistic effect, and even its compatibility later on in the extraction process and formulation by highlighting their physicochemical properties, such as whether they are water-soluble or soluble in organic solvents. Having knowledge on these different metabolites, properties, and characteristics is very important from raw material processing until product formulation. And Herbanex laboratory processes are considering all of those. The complexity of constituents of herbal extracts is complicated but can be predictable. Again, thank you, Sir Aidan. And for our participants, I encourage you to type in your questions in the Q&A panel, and it will be entertained during the open forum session. Again, thank you, Sir Aidan. All right. For our next speaker, she earned her bachelor's degree in chemistry from Colegio San Agustin, Bacolod in 2015. She is currently the Analytical and Laboratory Services Head of Herbanex, which caters to natural products researchers from all over the Visayas. She has worked as an RA under the PCHRD Drug Discovery and Health Product Program from 2016 to 2019 on dosage form development and standardization. She wants to pursue natural products research with specialization in drug discovery and natural product synthesis as her long-term career. She will be giving a lecture on standardization of extracts and dosage forms. Please welcome Ms. Herian Marie Ibanez. Take over, Ms. Herian.
Thank you, Claire, for the introduction. Let me just pull up my screen. So, is it okay? The screen. So, good afternoon and hello, everyone. I will be giving a short talk on the standardization of extracts and dosage forms. Okay, so standardization, this is the process of prescribing a set of standards or inherent characteristics, constant parameters, and definitive qualitative and quantitative values, which would carry an assurance of quality, efficacy, safety, and reproducibility. Okay. The American Herbal Products Association has defined herbal product standardization as the body of information and controls necessary to produce material of reasonable consistency, which is achieved through minimizing the inherent variation of natural product composition through quality assurance practices applied to both the agricultural and manufacturing processes. So we can see in this definition that standardization goes hand in hand with quality control practices. And then in addition to that, according to the World Health Organization, herbal product standardization is the process involving the assessment of safety, efficacy, and stability of finished products, the documentation of experience-based safety and risk, and the provision of product information. Now, in the herbal product standardization, the term standardized can refer to any of the following. It could be a product manufactured to ensure that a given marker compound or marker compounds meet a specified level, or a dosage unit containing a specified amount of herb, or a consistent manufacturing process. But beyond the manufacturing processes, standardization can also extend to industry-wide acceptance of common names of herbs sold in the market, the safety labeling for commercial herbal products, and the validated analytical methods used to test the products. Now, traditional practitioners use herbs mainly in their whole form, and a whole herb or a raw herb or crude herb can be obtained from drying and grinding processes only, so they are expected to retain the full spectrum of natural compounds originally found in the plant. We see these often in the form of a plant powder, and although plant powders contain beneficial compounds, they also contain a considerable portion of inactive compounds. And so in order to be able to pull out and separate active plant metabolites from the inactive portion of the whole herb, extraction processes were developed, and there was a shift from the use of plant powders to the use of plant extracts or plant concentrates. It is also important to note that a certain extraction process is only able to pull out some compounds, which means that some bioactives may be left behind in the extraction process. Okay, just a review on the types of extracts which we can see in the market. We can classify the extracts in terms of their chemical composition and in terms of their physical properties. So under chemical composition, we have the galenical or the full spectrum extracts, which are termed so because they are extracts which most closely represent the chemical complexity of the plant material. These are obtained by mixing various extracts together. And we also have the selective extracts, which are obtained by using a very select extraction process for solvent. Now, in terms of physical properties, we have our liquid extracts. Under liquid extracts, we have the tinctures and the soft extracts. Okay, and these extracts have a DER or drug equivalent ratios of 1:10 or 1:5 and below, which means that 5 liters of the extract is obtained from 1 kilogram of the herb. Also, extracts are available in semi-solid or solid forms, and these have, or in spray-dried and freeze-dried form, these have DRs of 2:1 to 25:1, which means that 1 kilogram of the extract was obtained from extraction of 2 kilograms up to 25 kilograms of raw herb. Now, the importance of drug equivalency or drug equivalent ratios is that we can use it when comparing the dosage of an herbal preparation against traditional literatures and other medicines. Now, equivalency implies that another preparation of an herb, such as an extract, is still suitable, provided it is declared equivalent to a dosage of a dried plant material. Now, herbal medicines derived from plant powders or plant extracts, they account for a large percentage of the pharmaceutical market, but a number of these herbal preparations suffer from a lack of standardization, especially on the quality control criteria for the raw materials, for final products, as well as for the processing methods. And this is where the substantial variations in compositions and quality of herbal preparations arise. And in order to address or minimize those variations and to ensure that consumers are provided with high quality botanicals, standardization has to be carried out. Now, because it provides products of reasonably consistent composition for the conduct of reliable clinical trials. It also allows the use of a positive control for the manufacturing process and allows dosage form manufacturers to confirm that the correct amount of extract is contained in a dose. And these substantial variations arise from the following factors. We have first the raw herb quality, which naturally vary according to the plant source, plant genetics, the environmental factors, and harvesting age. And then those which affect the quality of herbal extracts primarily are the specific plant parts used, the post-harvest processing, which includes the drying, transportation, and storage parameters, as well as the extraction methods. So later we'll be expounding on that. And all of these result to differences in levels of biologically active compounds and contaminants, both biological and toxicological in nature, in your herbal preparations. So next is, how do we go about standardization? So first, there is a need to review pharmacognostical references. So we have our pharmacopoeias, our pharmacognostic literatures, which are available for the specific herb under study, and being able to review these helps us to lay down standardization parameters, which may be structural or physical, chemical or biochemical, and biological in nature. And also, it helps us to lay down the methods for ensuring plant identity, detection of adulteration, and as well as identify mechanism of actions for herbs. Because this would serve as the basis for the conduct of preclinical and clinical studies for demonstrating the effects of the herbal preparation. Now, standardization also requires the careful control of raw material quality and during all phases of the manufacturing processes. This is a very important prerequisite step to obtain preparations of consistent quality. We have to be aware that the various activities of herbs depend upon the multiple chemical components and their synergistic or antagonistic interplay. Now, the primary active components of many herbs have not been completely identified, and focusing on the known active or marker compound on this stage of the standardization work may result in a reductionistic view that the herb's activity and benefits depend solely on that compound. Therefore, instead of the conventional single compound approach, pattern-oriented or fingerprint-oriented approach should be used in quality monitoring of raw materials because it makes use of all the components at the same time, even though their chemical properties may not be fully known. Okay, so where chromatography is used, for example, as the main tool for getting the profile of the herb, it is important that the profile generated must be comprehensive enough to provide a basis for assuring identity and consistency. Now, the generated profile chromatogram should aim to reflect the possible variation, including the potential adulterations and substitutions which may occur for the herbal material. And chemometrics data processing methods have been developed and integrated to fingerprint-oriented approaches to improve quality monitoring. These approaches enable fingerprint models to be produced and getting a thorough profile of the herbs, for example, a combination of its taste profile, mineral profile, chromatographic, phytochemical, and biological profiles or macronutrient profiles. This would give us a very large data set. Okay, so in order to interpret such data sets, data processing methods are required to reduce their dimensionality, but in an interpretable way, at the same time without losing important information. So one of these tools or data processing tools is the principal component analysis or PCA. Okay, so PCA at this stage of the standardization work helps us define which variables are responsible for the certain classification of the different accessions. Okay, so in selecting possible sources or discriminating other varieties of raw materials, chemometric-based approaches are really helpful. So this step can be done at the laboratory scale before scaling up. Okay. Now, in the production environment, we know that variations of some degree will occur on a batch-to-batch basis, even after minimizing the variability in the raw material. This can arise where the extraction processes are not standardized or are not or continue to vary for the same raw material. Now, extraction processes, these largely influence the quality and spectrum of components of an extract, and these should be optimized and kept constant. Now, these parameters include the particle size of the powder, the solvent type, solvent strength, and the number of extraction cycles and the duration of the extraction process. Now, once extraction process has been optimized in the laboratory scale, a fingerprint for the extract can be established, and this fingerprint can serve as the positive control when scaling up the extraction process. So, for example, in our standardization work with DDHP, we have set 80% ethanol as our default extraction solvent because it is able to extract a wide range of compounds of different polarities. Now, as standardization in the industry is really market-driven, we also found it important to have a standardization target and optimize our extraction parameters based on that selected standardization target. So, for example, this is one experiment where we had to validate the use of 80% ethanol as a solvent for Centella, or if you're familiar with that, that is Gotocola. So these are the TLC chromatograms for Centella in various ethanol concentrations as well as in water. Okay. So we found that 80% ethanol did contain the highest amount of the 4 triterpenoids. So the market for Centella is mostly for the treatment of venous insufficiency, and that these 4 triterpenoids are already well-established markers, and a wide range of products use this as their standardization target. So we optimized our extraction solvent based on the total of the 4 terpenoids in Centralia. Okay, this is not the same as saying that this compound or group of compounds is solely responsible for the activity. They don't necessarily represent the therapeutic activity, but having a standardization target also helps us to ensure that the extract or the preparation, which we would be incorporating into a dosage form, for example, is of a consistent quality. It should also be noted that a compound or group of compounds may be chosen as standardization targets only when they are already known and are already well established. Now, we had another experiment where another herb which we had to standardize was banaba. So we used chlorosolic acid primarily as our basis of standardization for the extract. Since we found that chlorosolic acid level is high in 80% ethanol, we opted to stick with 80% ethanol as our extraction solvent. However, the water extract is also reported by a lot of authors to have the antidiabetic properties. We were also able to produce the water extract wherein we used ellagic acid as a secondary marker. So for banaba, we had 2 different extracts which we later incorporated together into one dosage form for diabetes. But the dose is still based on chlorosolic acid. Excuse me. Okay, so still on the optimization of the manufacturing processes, the extracts and dosage forms should also be standardized by monitoring not only their chemical composition, but their physical properties as well. So part of the standardization work is to optimize powders, granules, capsule, and tablet formulations. The excipient type and level, as well as the granulation processes, can affect physical properties such as compressibility, flowability, and hygroscopicity of powders, granules, as well as the hardness and friability of tablets, and so on. So the next step is to be able to establish the quality control criteria for the preparations. This is done by subjecting a number of production batches to statistical analysis in order to establish an acceptable range of quality for the herbal preparation. So this process is called validation. The consistency of herbal preparations also have to be proven through the use of adequate test methods. And these test methods also need to be validated. And that includes validation of chromatographic and spectrophotometric methods where applicable. Okay, so these are just some of the quality parameters which are basically monitored for herbal preparations. We have the structural, physical, chemical, biochemical, and microbiological parameters for the powdered herbs, powdered extracts, and dosage forms. These same parameters should also be monitored when conducting stability studies. So in our standardization work, we use HPLC as our main analytical tool for the qualitative and quantitative chemical analysis. This table is just a comparison of HPLC and HPLC. So the HPLC as a technique is very simple and allows for shorter analysis time. Although HPLC can give relatively higher resolution and has relatively higher sensitivity, HPLC is already a well-established technique and has great application in qualitative and quantitative analysis. Now, if you remember the definition, or according to WHO or the World Health Organization, they have mentioned that standardization should also include the assessment of safety, efficacy, and stability of the finished product. Aside from the obligatory requirement in the registration process for all medicinal products, stability testing is important to know that the biological contaminants, chemical composition, and activity of the herbal product is not significantly altered. So assessment of the toxicity and efficacy is also a very important step. And some efforts have also been made in putting both chemical and bioactivity information together, as these may prove useful in stability studies too. So for example, an herbal preparation with anti-diabetic function to have its alpha-glucosidase inhibitory activity monitored along with the other physical, chemical, and biological parameters during stability studies. And then the last step is to be able to assemble a monograph for the herb or the herbal preparation, which, based on the World Health Organization, should also be included in the standardization work as part of the provision of information on herbal products. Now, monographs should be able to contain basic descriptions for the herb, including its nomenclature, the plant parts used for the preparation, the constituents of the plant, the range of applications, the dosage, and the mechanism of action of the herb, as well as a compilation of the identification tests and assay methods. So that ends my presentation for today. Thank you very much for your attention.
Thank you for that very informative and interesting talk, Ms. Hurrian. To our participants, perhaps the variations in every extract you obtain, especially with regards to the quality, have been your concern for some time. We have heard earlier from the talk of Sir Aidan that we can make a drug out of the herbs. The herbal medicines, however, suffer from lack of standardization parameters. And usually the main limitation is the lack of standardization of raw materials, processing methods, final products, and even the non-existence of criteria for quality control. Based on the presentation of Ms. Hurrian, I believe standardization of herbal drugs is not an easy task, as numerous factors influence the bioefficacy and reproducible therapeutic effect. But having a technology and set of approaches or procedures and optimization process at Herbanex can make the work much easier. Thank you for sharing to us your works at Herbanex, Ms. Hurrian. And I think we already have here a question from one participant. Ms. Hurrian or anyone from the team will answer it during the open forum. And for other participants, you may also type in your questions in the Q&A panel at the bottom of your screen. Again, thank you very much, Ms. Hurrian. Now, moving along to our session, we have our next speaker. She is a licensed chemical engineer who graduated from the University of St. LaSalle in 2018. She is currently the supervisor of the Applied Research and Innovation Laboratory, or ARIL, of Herbanex Incorporated, and also works as a research assistant under Herbanex DOST-BIST project. She specializes in process optimization and the development of semi-purification methods for ingredient manufacturing. A writer on the side, she wants to pursue bioengineering and apply her chemical engineering training into in situ resource utilization to help pave the way for human space explorations in the future. Wow. So let us welcome Ms. Andrea Nicole Farrell as she gives her presentation on raw material processing and ingredient manufacturing. The floor is yours, Ms. Andrea.
Thank you, Ms. Claire, for the introduction. Let me just pull up my slides. So good afternoon, everyone. Hope you are all doing well, and thank you for joining us today. So to proceed with our webinar, I will now be discussing the raw material processing and ingredient manufacturing process that happens at ARIL. So as mentioned by the other speakers, herbal products have increased in importance in the treatment of health-related problems, causing a constant and at times exponential growth in global interest in the use of herbal medicines. So the extractive isolates of herbal products are extremely complex multi-component mixtures, as opposed to synthetic drugs that are most commonly a single pure compound. And for this reason, the herbal products should be made of raw materials required for quality, because only then they could be safe and effective. However, of course, nature does not supply its products with a consistent, standardized composition. The constituents of medicinal and aromatic herbs can vary greatly as a result of multiple factors. So this high variability found in raw materials is one of the main problems related to quality, which is why raw material processing and ingredient manufacturing are crucial processes in product manufacturing. Here is the general process flow that we follow in ARIL in the manufacture of spray-dried extracts, which could be used as active pharmaceutical ingredients. Now, I will be going through each of these steps and discuss what exactly happens here and their significance. I would also be giving examples and insights from our experiences in-house. So first is raw material processing. Now in herbal processing, this is referred to as the primary processing step, which involves post-harvest treatments that free the herbs from foreign matter and other contaminants. This includes your sorting, your washing, and your drying. So other herbs may actually require further processing, which is why this is called primary, because there's actually your secondary and your special processing. For example, to improve flavor of one of our manufactured teas, we ferment it as a secondary processing step. So to start, we have sorting, which is the first step to ensure purity and cleanliness of your plant material. This involves removal of dirt and foreign substances, discarding of your damaged parts, and separating of unwanted plant parts. Next is washing, which removes remaining impurities. So in the past, we were manually washing herbs, but with large quantities, it is more efficient and cheaper to use machinery, such as your mechanical washers. Care must also be taken to avoid soaking your plant materials for unnecessarily long periods of time, to avoid loss of your water-soluble phytochemicals. So what is the significance of sorting? Adulteration commonly happens wherein your foreign materials are mixed in the herb supply. Suppliers often do not have stringent measures to prevent adulteration. So it is the responsibility of the manufacturer to sort the herbs received. So common foreign materials are listed here, and they're often mixed in during the collection or harvesting process. For example, in figure 1, you can see that hydrocotyl ambiliata is often misidentified as Centella asiatica due to their similar appearance. In figure 2, two medicinal plants, Videns pilosa and Tridax procumbens, are also often interchanged. And difference in quality in one batch, wherein some herbs are fresher than others, can be observed due to post-harvesting processes. You can see this in figure 3, which shows one batch of tawa-tawa samples with the freshest one in the right side. So, sorting enables the manufacturer to choose the herbs deemed still in good condition to include in the raw material to be processed. So, by sorting your raw material, not only are you preventing adulteration and contamination, you're also helping to reduce your unnecessary load to your subsequent processing methods. After that, drying is done immediately after sorting and washing to ensure good keeping qualities and reduce damage from molds and other microbial infestation and stop enzymatic action, which can alter your phytochemical content. It also facilitates your grinding process and subsequently your extraction process. So drying conditions are determined by the nature of your material and the desired appearance of your final powdered herb. It is also dependent on character of your active ingredients and the texture of the plant part collected. So generally, compounds in herbal medicine are relatively stable, except for your volatile compounds, your enzymes, and some vitamins such as vitamin C. So volatile compounds commonly evaporate at temperatures around 60 degrees Celsius. So proper drying involves the control of your temperature, humidity, and airflow, and other parameters. So final moisture content depends on the tissue structure of herb, but generally it is observed to be below 12%. However, since volatile compounds can be lost in drying, optimization of your process conditions is a must to ensure good quality of the resulting dry material. So what are the drying process variables that we can control? First is the drying temperature. As we know, heating can increase your free radical scavenging activities and inactivate enzymes, resulting in higher TPC. But it may also degrade your heat-sensitive bioactive compounds and cause the loss of your volatile compounds. Also, it's your drying time, which is usually linked with your drying temperature. Excessive drying times can degrade valuable compounds and unnecessarily increase your energy consumption. So we can observe the TLC profiles of the dried herbs to determine if a change is caused by the temperature or the time of drying. So for example, in an in-house experiment, we observed that ginger rhizomes, when dried at 50 degrees Celsius, had 1.59 times more 6-gingerol content than rhizomes dried at 60 degrees Celsius. So other parameters include your load and your air velocity, which can affect the drying rate or evaporation rate. So what do you use? Is it high temperature or is it low temperature? To help us choose, we consider the effect of each condition to the quality. So for high temperatures, you should know that it enables rapid drying, especially for large quantities, but it may also result to loss of aroma and possibly your heat-sensitive compacts. Meanwhile, low temperature drying reduces the loss of those volatile compounds and heat-sensitive compacts, but prolonged drying time may result in darkening of color and possible increase in microbial load. So after your drying, we proceed with grinding and sieving. So the purpose of powdering the herbal material is to rupture its tissues and cell structures so that the chemical ingredients are more readily exposed to the extraction solvent. Also, size reduction increases the surface area, which enhances the mass transfer of your chemical ingredients to the solvent. However, desired particle size range is required due to the fact that an excessively fine powder may affect filtration during extraction, and it may even coalesce in the presence of the extraction solvent to form solid lumps, cakes, or bricks, which is not amenable to being extracted. Hence, your optimum grinding size is dependent on the nature of your raw material and the succeeding extraction methods that will be used. So some key considerations are included in the table shown. So if your extraction is agitated, then it is okay to use smaller size particles, since agitation will prevent lump formation. Meanwhile, if you are doing multiple extraction, then it is fine to use larger size particles. Starchy materials also require to be larger in particle size since they tend to become sticky when contacted with the extraction solvent. So after that, and you are able to produce your powdered herb, we go on to ingredient manufacturing, in which the first step is your extraction. The purpose of extraction of herbal material is to eliminate your unwanted materials and to concentrate other chemical constituents in soluble form. So some of the extraction methods available at Arel include your decoction, which is what is traditionally done for the preparation of your medicinal herbs. In Tuplas Lunas, we usually do extraction by maceration, which involves mixing the properly comminuted herbal materials with a solvent and allowing the mixture to stand at a certain temperature for a defined period of time, agitating if necessary. A new technology is subcritical fluid extraction, which uses solvents such as your water or butane at subcritical conditions. It has the advantage of being able to extract lower operating temperatures and pressures, environmental compatibility of your solvents, good selectivity, and low residual solvents. It has excellent dissolving power for lipophilic compounds, which is why this technology is used to extract oils by companies abroad. In the lab scale, we offer your Cyclotide extraction, which integrates the advantages of your reflux extraction and percolation by continuously extracting the herb with fresh solvent. And lastly, is your sonication, or specifically ultrasonication, which uses the application of ultrasounds to form cavitation bubbles throughout your solvent. And that collapse causing pressure and temperature changes and therefore enhancing the rate of mass transfer. So how do you select the proper extraction method? You need to consider the nature of the material, its stability, its solubility, etc., and the amount you have to extract. Your method should also be relatively rapid and exhaustive, which means you're able to remove as much as of the desired chemical constituent as possible from your plant matrix. And the method should be simple, scalable, economical, environmental friendly, and reproducible. So what are the process variables that we can control during extraction? First is your extraction solvent, which the other speakers had mentioned before. So some of the considerations you have is the solubility of your target compounds, stability, and reactivity, and the others. You also have your temperature. You have to consider the heat sensitivity of your target compounds. And then there's your extraction time. What is the rate of mass transfer of your target compounds, your solvent? And is your active phytochemical constituent sensitive to prolonged extraction? And lastly is your number of extraction cycles and your herb to solvent ratio, which considers the solubility of your target compounds in your solvent. So from your research, you already have a process flow of your own production methods. However, laboratory scale procedures are not ideal to directly translate to commercial scale, as mentioned before by Sir Philip. So technology transfer involves consideration of equipment capabilities and costs that will ensue. So, which is why here at ARIL, we offer process optimization, which is the development, decision-making, and improvement in the chemical processes in order to maximize your process performance and minimize your processing costs. So, for example, you want to improve your product yield, your product quality, your throughput rate, your product purity, and improve your microbial load. And you want to reduce costs such as energy, labor, raw materials, equipment, materials recovery, and your waste processing costs. So here at ARIL, we conduct two types of optimization. First is your single parameter optimization, which is what is traditionally done. And only one factor is considered at a time. And then there is your response surface methodology, or your RSM, which explores the relationship between several process factors and one or more response variables. So this method is more efficient than your traditional single parameter optimization because from a limited number of experiments, RSM generates various model graphs from the data you obtain, which helps you locate the regions where the response you want is optimized. So some common responses that we measure are your TPC, your TFC, your antioxidant activity, and we use your TLC quantification of markers and profiling. For example, in single parameter optimization, we tested the extraction of OR status or your cat's whiskers by trying different solvents, such as methanol, water, and different concentrations of ethanol. So as you can see in figure 4, the relative yield of synensitin, or your water marker, is observed to be high in ethanolic extracts, increasing with increasing ethanol concentration, and the highest at 97% ethanol. Meanwhile, water is shown to be the least suitable solvent. Another example, but for RSM, is shown in figure 5, which is a 3D response surface plot for the effect of extraction time and temperature on the response, which is your total soluble protein content. So this is only a part of the results, and the model actually generated was found to be quadratic. So from these results, we could infer that thermal denaturation is thought to be the primary reason of the loss of protein content at higher temperatures. So after your extraction is your concentration. So before spray drying, we concentrate our extracts to recover the solvent and to reduce the volume of your extracts, and subsequently reducing the energy required for your spray drying. Typically, the extract is concentrated to 20% of the volume. However, for some herbs that produce viscous extracts, this is not the case because a high degree of concentration is not possible. We want to avoid excessively concentrating our extracts in order to avoid also creating excessive viscosity, because viscous feeds require more energy to pump and can be difficult to atomize in the spray dryer. Also, another thing to note on is that your volatile oils could actually be removed during your concentration process. Should these oils be the key to the bioactivity of your extract, it is actually possible to capture them, but with the proper equipment. Hence, the equipment utilized is an important consideration in concentration. After concentration, there may also be a need to add excipients. After all, your crude extracts are often difficult to spray dry due to the presence of undesired compounds, as mentioned by Sir Philip before. And because of this, these compounds make processing difficult by lowering your yield or producing SDEs that are hygroscopic or poor in flowability. And so your excipients are added to improve the manufacturability, the flowability, and the stability of the product. So, for example, in figure 6, you're showing the effect of excipients in the spray drying of your turmeric extract. Different combinations of different types of excipients were used. As you can see, low yield is observed for some combinations due to the SDE sticking to the walls of your spray dryer. Meanwhile, maltodextrin at a certain percentage is shown to give the highest percent yield out of the excipients used. So it depends upon your raw material. After concentration, we then proceed to your spray drying, which is a unit operation wherein the liquid feed is sprayed through a hot gaseous drying medium, which is usually air. So it has been employed in a great number of industries for purposes such as dehydrating your products, aiming for preservation, ease of handling, transport, and storage. It's also used to encapsulate bioactive compounds within a protective matrix or to induce phase transformation of solids to obtain dried powders with distinct properties. So spray drying is considered a powerful technological process since it brings visibility to the production of free-flowing particles with well-defined particle size. So how does it work? It involves 3 steps. First is your atomization, wherein your liquid feed is dispersed as droplets to increase the surface area and as such optimize heat and mass transfer. Then these droplets are contacted with your hot gas, which results to conversion into particles. And lastly, these particles are collected by the bottom of the chamber or with external devices. So here is a table which compares the 3 common drying methods used. For manufacturing, spray drying is preferred due to its advantage of being a rapid, continuous, and single-step method that displays great versatility and reproducibility. As you can observe, freeze drying may prevent the oxidation damages and minimize chemical compounds change, but it requires very high facilities and a cost, and it is a slow and expensive process. So this is why spray drying is preferred in the industry. And the process variables that we are able to control during spray drying include your atomization pressure or atomization frequency. This depends on the atomizer type used, your feed flow rate, your feed viscosity, your inlet temperature, and your outlet temperature. Of these parameters, the most important are inlet temperature and your feed rate. So by developing the optimum process conditions for each herb, we are able to create spray-dried extracts with good quality fit for API use. Before I end, here are some remarks on microbial load control. Herbal raw materials contain high levels of microorganisms, which could be potentially pathogenic to man. Hence, guidelines for permissible microbial load exist for the registration and approval of herbal food supplements and medicine. So I included here 2 guidelines, examples of guidelines, and only the total aerobic microbial count. In ASEAN, as you can see, it here is the TAMC limit required, while in the DOH Administrative Order 172/2004, it is different for different materials that will undergo different treatments and purposes and the type of the product. Where powdered herbs are used, as in the DDHP herbs such as sambong and lagundi, the practice here in the Philippines utilizes gamma irradiation, as offered by the Philippine Nuclear Research Institute. At ARIL, it's difficult to send all of our raw materials to PNRI, and Urbanex is organically certified. Hence, we utilize other decontamination measures such as your high temperature, short time sterilization, and other proprietary technologies that we are currently developing. And speaking of developing technologies, here are some of the other methods that we are currently exploring. We are developing heated ethanol extraction, as heating is shown to aid the extraction of phytochemicals while reducing the extraction time that maceration usually requires. And under the DOST BIST project, for which Urbanex is the first grantee, we are developing semi-purification methods to improve the quality of STEs and reduce the need to use excipients. So through this project also, we have been able to procure equipment to further our research on API manufacture and also establish the Applied Research and Innovation Laboratory, or ARIL. And that ends my presentation. Thank you for listening. I hope you were able to get valuable insights from my talk, and it has guided you in what to expect when you go into product commercialization. So for any questions, kindly reserve it for the open forum. And I hope you can scale up with us. Thank you and good afternoon.
Okay, thank you very much, Miss Andrea, for generously sharing to us the processes in preparing high-quality raw materials at Herbanex Laboratory. Thank you also for showing to us the actual pictures on how the raw material processing and ingredient manufacturing are done in the lab, as well as their optimization processes, which you can offer to your collaborators as well. Well, I believe you earned our confidence in having a raw material that is of high quality, which will directly impact the safety and efficacy of the final product. No doubt, Herbanex is rising fast because of competent staffs. Again, thank you, Miss Andrea. All right, everyone, it was just an hour and more only, but we were loaded already with information. I guess by this time, you are already thinking of projects you can work at Ariel. To refresh ourselves, let's have a 5-minute break from our talks, and you may watch the video presentation about Ariel by Herbanex Laboratory Incorporated. Thank you. By the way, just to share with you, I was an intern of Herbanex Laboratory, I think year 2014. And during that time, there were only few equipment present in the lab. So having this virtual tour of Ariel made me proud even more to Herbanex Laboratory. Although I wasn't surprised at all that in a short span of time, there were big improvements in the lab already because of the commitment of Sir Philip Cruz and the rest of the Herbanex team to level up the herbal industry in the country. To be honest, as a pharmacist, I have a low confidence to herbal products in the market because of issues on safety and quality. So having this laboratory and vision, I am excited to see more herbal products that will become beneficial to Filipino people. Again, thank you for the relaxing video presentation, Herbanex team. Kudos to everyone. Now, down to our last three speakers to talk about product development, we have the following speakers. Our first speaker in the product development topic is a graduate of food technology from the University of the Philippines in the Visayas in 2016. She is currently the product development head of Herbanex R&D unit. Her work focuses on the formulation of food supplements and functional food products, expanding application of Philippine medicinal plants for health and wellness. She loves to hike and wants to someday contribute to research initiatives in food sustainability. And for our second speaker for product development topic, she is a graduate of BS Pharmacy from the University of San Agustin in Iloilo, where she used to be an instructor. And she was my instructor before during my college years. She is currently taking up her MS in Pharmacy at Central Escolar University, Manila. She has experience in herbal drug manufacturing R&D and in quality assurance. She currently heads the pharmacy internship program of Herbanex, where she nurtures and mentors pharmacy students, especially about industrial pharmacy and herbal medicine development. Let us welcome Ms. Nyambe Ruth Palacio, who will deliver a talk on functional foods and beverages, and Ms. Mary Colleen Ibanez, who will talk about drug and food supplements. Take over, moms.
Good afternoon, everyone. I'm Nyambe Ruth Palacio. I'm just gonna pull up my slides for you guys. All right. So good afternoon, everyone. We are coming to the last stretch of this webinar, so I hope everyone still has enough energy for a few more presentations. Ms. Colleen and I will be discussing on product development, and I will be focusing on the product development process for food products. Before we begin, I would like to present an outline about the topics that I will be focusing on today. I will be touching on product ideation, establishment of product specifications, product formulation, pilot scale manufacturing, and stability study. Just a brief overview. So this is our product development process flow. Product development begins with product ideation, then proceeding to establishment of product specifications, product formulation or lab scale production, pilot scale production, stability study, and finally the final product. I will be discussing each step along the way. So product ideation usually begins from the client or the researchers. Often the client wants to bridge a gap in the market. Here at Herbanex, our clients usually come to us wanting to develop food supplements. Each client varies depending on their target market, whether they are developing food supplements for young adults, lactating mothers, the elderly, etc. Then we get into technical and economic feasibility. Can the product be produced in large quantities? Do we have a sustainable supply chain system in place? And what would be the cost of the whole process? And can the client shoulder the cost until they have reached a return on investment? Lastly, what are the product requirements? Clients come to us with a specific herb or a combination of herbs in mind. Sometimes they're not sure about what ingredients to use, but know that they want to develop a tablet or a capsule. Once the client has communicated this with us, we proceed by establishing initial product specifications. Product specifications is the part where we offer technical advice and ask for more details of the product. Do they want their product in a capsule form, a tablet, a powder, a beverage? What would be the main ingredient they want to highlight? And is there a specific active compound they want to focus on? We provide suggestions based on experience on what type of packaging material we can offer, decide on the product weight or volume, and take into consideration of any additional quality requirements, such as the need to be vegan, halal, gluten-free, etc. Note that even though we have established these specifications at the beginning of the project, it is possible for the product to transform into a different direction later on as the development continues. And now we proceed with product formulation. I'm sure that every participant here is familiar with trial and error. Product formulation can be a simple or complex process. Product formulation can go from 2 weeks to up to 3 months, depending on the complexity of the product. By the end of the product formulation, we should be able to establish the final formulation, the manufacturing process, its technical specifications, and the containers and packaging materials for the product. For the purpose of this webinar, I will be focusing on food supplements. One of the considerations in making food supplements is choosing the active ingredients. Currently, our products are made either from powdered herbs or herbal spray-dried extracts. Each ingredient has its own advantages and disadvantages. Powdered herbs are commonly used in many commercial herbal food supplements, and it is easy to produce since all it requires is drying, grinding, and sterilization. However, due to the availability of new technology, we were able to maximize powdered herbs and turn them into spray-dried extracts. Although spray-dried extracts has a more complex manufacturing process, we found that these extracts have higher bioactivity than powdered herbs. As discussed in the earlier presentations by Ms. Hurian and Ms. Andrea, spray-dried extracts can be standardized. However, working with extracts also presents with its own set of challenges, such as finding the right excipients and granulation solvents to work with. So these are considerations in making a food supplement. Presented in this table are the basic ingredients we work with in developing food supplements. So first are excipients, which are often called as carriers or binders for people who are in the pharmaceutical industry. When working with excipients, we first consider the characteristics of the main ingredient. Does the excipient result in good granule size? Does it help us reach our desired dose per capsule? And in hygroscopic raw materials, does it help address that problem? Examples of excipients that we work with here at Herbanex are maltodextrin, lactose monohydrate, mannitol, and pre-gelatinized starch. Another consideration are lubricants. They improve flowability in powders, which is important during dispensing during production. A few examples of these are magnesium stearate and silicon dioxide. Lastly is the granulation solvent. We often use water or alcohol as granulation solvents. Considerations include the raw material solubility in water or alcohol and the quality of the wet mass produced during wet granulation. On this slide is an example of a standard granulation process. Dry ingredients are sieved and compounded before the addition of a granulation solvent. This produces a wet mass, which is then sieved through a mesh before being dried in an oven. Once the granules have reached the standard moisture content, granules are passed through another sieve in a process called dry granulation. The lubricant is then added with the dry granules for final blending. Granules are then capsulized or tableted into the final product. Our food supplements often come in two forms, namely capsules and tablets. Each product form has its own set of advantages and disadvantages as presented in this table. Coming to a decision often narrows down to the raw material, the product application, and the cost of the product. Each product's technical specification is established during product development and is based on existing regulatory standards. So when we say technical specification, it refers to the product's characteristics at its ideal or most stable form. In order to create a product's technical specification, it must undergo a series of tests to evaluate the product. It is in these tests where we get additional information on how to further improve the product, such as substituting existing ingredients, a change in the manufacturing process, etc. On the table presented on the left is a list of tests we use to evaluate granules, capsules, and tablets. Data acquired from these tests help us evaluate the overall quality of the product, as well as guide us on how to make adjustments in the formulation. So I will just focus on a few technical specifications. So one of these that we're having issues with or having challenges with is hygroscopicity. So working with spray dried extract means that the powders are very hygroscopic, and we have to adjust our formulations in order to preserve the active compounds and at the same time ensure that the products are stable. So once we have evaluated the technical specifications of the product, it is time to identify which packaging material to utilize for the product. So currently at Urbanext, we offer the following packaging materials: strip foil, blister foil, and PE bottles. Next is the pilot scale manufacturing, or commonly known as process scale-up. The process scale-up evaluates the replicability of the manufacturing process from the laboratory scale to the production scale. In this step, we determine the manufacturing specifications and establish critical process parameters. Pilot scale production is the transition stage from lab scale to industry scale production. It helps us determine a product's equipment requirement. If you look at the photos on the right side of the slide, you will see two types of granulators, the extruding and the oscillating granulator. In a pilot scale process, we tested out these two equipment and evaluated which produced the best granules for this specific formulation. Oftentimes, the behavior of the formulation changes at higher volumes, and we have to adapt to the process that best replicates our data from the lab manufacturing scale. Second is scaled up manufacturing process refers to the entire process flow from beginning to end of production. This includes the ingredient dispensing to final packaging. Next is quality specification. So this refers to quality standards established for the product during and after production. This includes the product's physical, chemical, and microbiological properties. And last is the manufacturing cost. Since the pilot scale production utilizes production scale equipment and estimate production time, it gives us a clear idea on manufacturing cost. Another importance of pilot scale production is establishing critical process parameters. This allows us to ensure replicability of results from the lab scale to the production scale, ensure compliance with good manufacturing practices, create foresight on possible risks and hazards throughout the production process, and provide appropriate protocols to address them. Last is to ensure that both of the product and the production process meet regulatory standards. Our quality control standards are based on internationally recognized organizations and local regulatory bodies. So the organizations presented on the right side of the slides are samples of organizations that we base our standards from. Another step in product development is the stability study. So the main objectives of a stability study is to select adequate formulations and container closure systems to determine shelf life and storage conditions, to substantiate the claimed shelf life, and to verify that no changes have been introduced in the formulation or manufacturing process that can adversely affect the stability of the product. Samples for a stability study are taken from a pilot scale batch, a production scale batch, or a combination of both. Samples are used for the stability study must have the same manufacturing process, formulation, dosage form, and container closure system as the final product. There are two types of stability studies. One is the accelerated stability study, and another is the real-time or the long-term stability study. Accelerated stability study are stored at heightened temperatures, specifically 40 degrees Celsius and 75% relative humidity. It lasts for a duration of 6 months and is conducted for registration purposes. It is conducted in increased rate of chemical and physical degradation to predict long-term effects and evaluate short-term exposures outside the labeling storage conditions. Over the course of the 6 months, the WHO suggests that at least 3 testing points, specifically during the 0th, 3rd, and 6th month of the study. However, the product developers may opt to add additional testing points as necessary, and because products are stored at higher storage conditions, hygroscopicity of ingredients and its sensitivity to heat must be accounted for. Long-term stability study, or also known as real-time stability study, is conducted throughout the product's expected shelf life. The long-term stability study represents a more accurate description of the product's actual shelf life, and the study must have at least 8 testing points, namely every 3 months during the first year and every 6 months during the second year. Should the study extend for more than 24 months, products should be tested annually thereafter. Since the study lasts for a minimum of 24 months, it is often conducted in parallel with an accelerated stability study to provide initial shelf life data for registration purposes. So evaluation of results, a tentative shelf life of 24 months may be established provided that the active ingredient is known to be stable within the entire duration of the accelerated stability study. Should a significant change be observed at any point in the study, only the long-term stability study data will be considered as basis for shelf life. The degree of variability of individual batches affects the confidence that a future production batch will remain within specifications throughout the assigned retest period or shelf life. For registration of food products, accelerated stability studies suffices to justify product shelf life. To end my presentation, I would like to introduce Urbanex's product manufacturing capabilities. Here are examples of our existing food supplements. The first is the Daily Apple Tawa-Tawa capsules. These are the first Tawa-Tawa capsules made from standardized spray dried extracts. And to the right is the Perclans tablets made from turmeric, curcumin, and piperine extract. We also have a line of functional food products such as banana turmeric chips, dried sweet roselle, and roselle jam, among others. We also have a line of extruded corn snacks and instant cereal meals as well. We also have a wide array of instant beverages. Among our products include instant coffee, juice, or as we call it, herb coolers, and tea products. Most of our instant beverage products highlight local herbs such as chaanggubat, dalandan, roselle, and bignay, to name a few. Lastly is our cosmetics product line. This is a relatively new product development line for us. Among our products include facial toners, foot powder, creams, and soothing gels. We highlight the use of local herb extracts in cosmetics such as elemi, centella, citronella, and oregano. And that concludes my presentation on the product development process. Thank you so much for listening. I would like to give the floor to Ms. Mary Colleen Gomiaga to discuss the pharmaceutical product development in herbal medicine.
Hello, good afternoon, everyone. I am Mary Colleen Gomiaga Ibanez, a registered pharmacist, and I will briefly tackle pharmaceutical product development, including herbal medicine and some registration requirements for us to finally market the product. Okay, first, let us define some terms. Drugs. So we have articles recognized in official pharmacopoeia and formularies and are used in the diagnosis, cure, mitigation, treatment, prevention of human and animal diseases. Unlike food supplements that are not allowed to claim therapeutic use unless with proper justification. Medicinal products are drugs that are subjected to control under a health legislation. And then we have herbal medicine, which are medicinal products with plant materials as the active ingredient with specific claims. So it follows AO172, series of 2004 for the guidelines in registration. And this is where all the documentary requirements for the registration can be found. Next, we have traditionally used herbal medicine and are considered as pharmaceutical preparation, with which also contains plant material, but claims are based on traditional experience. It follows AO184, series of 2004 for the guidelines in registration. When registering a medicinal product, unlike food products or food supplements, more rigorous documentation has to be done. So it is necessary to plan out and align the product development of these products. For pharmaceutical or medicinal products, there are two guidance documents. So the ACTD, or the ASEAN Common Technical Dossier, for compilation of the necessary product information requirements. It has 4 parts: the administrative information, quality document, the non-clinical document, and the clinical document. The next, or the second guidance document, is the ACTR, or your ASEAN Common Technical Requirements, which is a compilation of the set of guidelines being followed by drug establishments. Included in the ACTR is the ASEAN Guidelines for Stability Studies, where, unlike food products or food supplements, requires results for both room temperature or long-term studies and accelerated studies. New chemical entities, as well as those that are known to be unstable drugs and dosage forms, requires a minimum of 3 pilot batches to be studied, while only 2 pilot batches are for stable drugs and dosage forms. When we say pilot batches, they are not commercial batches yet, but pilot batches where stability studies are taken should simulate the process of production batches that should provide a product of the same quality, meeting intended specifications. For the frequency of monitoring of stability studies for pharmaceutical products, long-term stability studies requires monitoring every 3 months for the first year, every 6 months for the second year, and yearly thereafter. While accelerated stability studies requires a minimum of 3 time point, which can be 0 month, 3 months, and 6 months. Results for both long-term and accelerated stability studies can already be used for registration, but a commitment to continue the studies is made to firmly establish the product's shelf life. Next is the ASEAN Guideline for validation of the analytical procedures which shall be used in the product. It was discussed by Ms. Heren Ibanez earlier in her presentation. It requires testing for LOD, LOQ, specificity, linearity, and etc. We also follow ASEAN guideline for the conduct of bioavailability and bioequivalent studies, which are applicable when preparations are not the innovator drugs or the first drugs for its claim. And the ASEAN guideline on non-clinical document or safety documents from preclinical trials, and clinical document for efficacy documents from clinical trials. Next is the ASEAN guidelines on the submission of manufacturing process validation data for drug registration. Under this guideline, 2 reports are needed. The pharmaceutics report, the development pharmaceutics report, which includes information about the product development, and the pilot batch manufacturing, which is supposed to be not less than 10% of the targeted commercial scale. This is done by a capable development party, Ariel, for example. The process validation protocol or scheme, which is done after the process development and pilot batch manufacturing for the purpose of marketing the product. This is done by the manufacturer itself in its manufacturing plant site after a technology transfer from the development party. The development pharmaceutics report on pharmaceutical development should address the following. So the rationale for selecting the dosage form, justification on the choice of liquid or solid dosage form, choice of product components. So we already want what our active substance would be, but we have to consider the excipients, the compatibility of these components, the physical-chemical characteristics, especially when using spray-dried extracts, as mentioned earlier, which are highly hygroscopic or has low flow property. Formulation of the products, the use of averages, the effect of pH, for example, its stability, homogeneity, and batch reproducibility considerations, the choice of manufacturing processes, like the kind of granulation and the drying, including sterilization procedures, choice of containers and packaging materials, especially if the product is moisture sensitive, the container closure integrity, or the absorption or leaching issues. Microbial attributes of dosage form, since lower limits are required for liquids than in solids. The compatibility of drug products with diluents. So this pharmaceutical development report should establish that the type of dosage form selected and the formulation produced or proposed are appropriate for the intended medicinal purpose specified in the application for drug registration. It shall also identify the formulation and processing aspects that are critical for batch homogeneity and reproducibility that has to be monitored routinely. It is important to note that this report is needed, the pharmaceutics development report is needed to provide a link to the process validation protocol for the manufacturer of the production scale or the 100% production batch size. An integrated approach to development, manufacturing, and quality for both industry and regulators is a concept called quality by design. It emphasizes that quality must be built in the product and will not improve by addition of testing or inspection. It is a systematic approach to development of a product with predetermined objectives and emphasizes product and process understanding, process control that is based on sound science and quality risk management. The mentioned predetermined objectives are in terms of defining the QTPP, or the Quality Target Product Profile. The QTPP is a prospective summary of the quality attributes or characteristic of a drug product to ensure the desired quality. By beginning with the end in mind or the target, the result of the development is a robust formulation and manufacturing process with an acceptable control strategy that ensures the performance of the drug product. It will provide an understanding that will ensure the quality, safety, and efficacy of a specific product for the patient. It will also serve as a basis for the development of the CQA. We will tackle CQA later, and CPPs and control strategies. Your QTTPs ultimately justify the choice of the dose, since that is your objective. So it will justify the choice of the dose, the nature of the packaging, the track, and the delivery device. The different characteristics of the product should provide an answer on the fate of the product after its administration to ensure its bioavailability necessary for its effectiveness and safety. And then we have CQA, or your critical quality attribute of the finished product. So these are the properties or characteristics that should be within an appropriate limit to ensure product's desired quality. These are the finished product's physicochemical and microbiological quality, and departure from these limits means an unsafe, non-efficacious, or low-quality drug product. During the development stage, the critical material attributes of the raw materials should be identified and should be monitored to be within the limit, as it can affect the quality of the final product. So we have to characterize our raw materials and identify the specific or the critical attributes of the material. And not only the material, but the processing parameters. It should also be designed in a way to identify the critical parameter to be monitored and controlled, again, to ensure the process produces the desired product quality. For the relationship of CMAs, CPPs, and CQAs, your input material or raw material attributes should be within the limit. The process parameters should be monitored and controlled in order for us to produce an output, a product of the desired quality, proven by the product's attributes that are within the appropriate or desired limits. Through proper formulation and process development, considering the CMAs and the CTPs, the desired product can be achieved to finally produce the clinical outcome for our patient. Finally, whenever there are changes, the CN variation guidelines includes the requirements as any change or amendment in the registered product must be approved by a regulatory body. There are two types of variations: the major variation, which has an impact on the product's quality, safety, and efficacy. And this is for the approval of the Food Drug Administration for the Philippines. And it requires certain conditions to be lifted and supporting documents to be submitted. Examples of major changes are the extension of using it for another purpose, changes in the product label, changes in the production or manufacturing site, more than 10% change in the batch size, and major change in the manufacturing process, since all of this can impact the product's quality, safety, or efficacy. There are minor variations that has no impact on the product's quality, safety, and efficacy. This is only for notification, but sometimes for the approval of the FDA. This requires conditions as well in supporting documents. Examples of minor variations are your change in test procedures, change in shelf life, change in specification of an excipient. So it is necessary to plan ahead if we are gearing towards the development of a food supplement or a drug product, and to plan with a target product in mind to properly address the documentary requirements and the guidelines to follow during product development. With that being said, I would like to thank you for your kind attention. Let's scale up and meet the market.
Thank you, Ms. Nyambe and Ma'am Colleen, for giving us more information on functional foods and beverages, as well as drug and food supplements. So what we have heard from our previous speakers before the break were not just theoretical or in the form of slides or presentation. Herbanex also offers tangible products that are manufactured using their technology and patterned with the requirements of FDA and ASEAN. I can see a strong quality control and quality assurance being practiced here. So Herbanex also scale up their lab scale research and turn it into a final product that is now marketed. Thank you, beautiful ladies, for your presentations. And for our last, but definitely not the least speaker for this afternoon, we have here with us Attorney Bayani Lustig. He is the Managing Director of Pinoy IP Works Incorporated, the intellectual property prosecution and valuation arm of Fortan Narvasa and Salazar. He is a certified patent agent and leads the intellectual property practice group of FNS. He heads the Patent and Technology Licensing Committee of the Licensing Executive Society, or LES, Philippines. He is likewise a member of International Federation of Intellectual Property Attorneys, or FICPI. He teaches IP fundamentals to engineering students at the Technological Institute of the Philippines, Quezon City. In his spare time, he tends to the coffee, cacao, and various Philippine trees he planted when the pandemic started. Attorney Bayani will give us a lecture on IP matters. Take over, Attorney.
Hello. Hello. Hello.
Yes, we can hear you, Attorney.
I think I can't. Thank you, Claire. Can you